I'm originally from Italy. I was born in Padua. When I was a little bit older, like middle school level, I started playing volleyball, and I kept playing in the team of the city, first in the junior team and then in the senior team. Then I kept playing in the national league till I was 25, and that was a big part of my life. I was playing volleyball professionally, which means like 7 hours per day. And then concurrently I was studying and taking classes, and that was a pretty intense phase of my life. Yeah, at that point I wasn't exactly sure what was going to happen, but I was sure that I really wanted to be a scientist. As far as I remember, it's my first dream job. When I started PhD is when I decided to quit volleyball. I had to make a decision whether to continue playing or to start being more serious about a career in science, especially in biology. While I was kind of thinking about what to do for the postdoc, I was really impressed by a few papers in the field of synthetic biology. I decided to join Wendell Lim lab at the UCSF, University of California, San Francisco, starting in 2012. And in this field, you kind of look at the cells like as little machines or little robots that you can try and program to make them do useful things. As a postdoc, I got very interested in how cells communicate to each other and in the possibility of engineering new ways in which cells communicate to each other. And one way in which cells communicate with each other naturally is by exposing signals on their membrane that are read by neighboring cells with sensors. That recognize specifically the signal, and once the sensor does recognize the signal, there is a message that is liberated from the surface, and it goes in the nucleus of the receiving cells, where it can change the behavior of the receiving cell. So what we wanted to do was to make the cells able to recognize different signals and being able to convey different messages. And the way in which we did it was to replace this part of the sensor with a different one with different specificity for the new ligand, for the new signal, and then modify also the message part with another part that would make the response different as well. And so now this signal of this cell can be a signal produced by a tumor cell, and so then the message could be killing of that tumor cell, or it could be a signal produced by a cell that's dying, a useful cell that is dying, and then the message and the response here can be something that can help the cell. I think that the capacity of increasing the control on stem cell behavior will be an area where synthetic biology can really help the field. I think it will be exciting to see how much we from synthetic biology can provide in forms of tools for the next generation of regenerative medicine therapies.